Okay, now I'm going to invite up our scientist speaker. Pallav Kosuri is an assistant professor here in the Integrative Biology Laboratory. He has done some amazing work here. He's also done a really cool video recently about the world's smallest Nerf gun. Yeah, so I think it's pretty cool. It's one of my favorite things ever. I nerded out so hard. But can we all give a big round of applause to Dr. Pallav?
Thank you. All right. Hi, everyone. I think this is the biggest audience I've had in this room. This is scary. Okay, let's go. Cool. Well, thanks, Shoshana. Thanks, everyone, for making this happen. I think this is wonderful, and it's the best reason ever for me to get up 2 hours earlier than I usually do on a Saturday. I'm glad to be here. I'm glad to see so many faces. And yeah, let's do some science. I'll start by telling you about how I got here a little bit, and then about one of the things I'm really excited about and why I get up early at 11 every Saturday to do science. Okay, let's start from the beginning. So I'm from Stockholm in Sweden. Hi, Mom. She's actually here. So I better behave. My mom is Swedish. My dad came from India originally, and then I grew up in Sweden. And I always dreamt of being driving one of those digging machines, maybe, or maybe being an astronaut or something else. I had no idea what I wanted to be, but I really liked machines and cool things. Eventually that turned into physics and mechanics, and I was like, yeah, people build really cool stuff. I want to build really cool stuff. And my biggest dream in high school and then in college too was there's this place in Switzerland where they build particle accelerators, and they literally build these giant circular machines that accelerate particles like protons and then smash them into each other to see what they're made of, to see quarks and things that nature is all made of. And my dream came true. I worked really hard, and then eventually I graduated with a degree in physics from a university in Sweden. And then I got to do my thesis project at CERN at this accelerator in Switzerland, and it was so cool. That's one of my buddies, right there. Look how big this machine is. It was so cool. And we built these giant machines to see the smallest things in the universe. I was really mind blown. This is another picture of, this one is called the compact muon solenoid because it's one of the smaller ones. But that's, so the world was spending, billions of dollars. This is, I think it's part of the largest machine ever built, actually. The Large Hadron Collider is the biggest one, but it's just all components in it. Anyway, it was mind blowing. But as I was there, I was like, is this really what I want to do? It is cool, but at some point I was like, okay, maybe other things are also cool, and maybe it's really interesting to know what nature is made of. But what if we can actually accomplish things also? Wouldn't that be cool? And I looked around and I was like, what's actually accomplishing things? And I realized humans are accomplishing things. And humans are so much cooler than these machines that we build. If you compare how advanced that machine is to how advanced this person is, it's just no comparison. Even if you take a finger of a person, is so much more complicated and intricate, and you cut it and it self-regenerates. How cool is that? That thing, if a cable came loose, we would have to search for hours and then try to locate the cable that was broken and then try to connect it again. It was pain. It was so annoying. And then every time we wanted to build another machine, we had to build a freaking other machine from scratch. And humans just build themselves. I was like, that's maybe there is a better way to do this. Maybe we don't have to build things. Maybe we can have things build themselves, I didn't make babies, but I realized that biology is so so awesome. Like, this is the nervous system of a developing fish. And this is not in real time, this is sped up, but you can see how this fish like literally builds itself, right? From instructions given in the genome. These are nerve cells growing into patterns in the body. This is exactly what happens to us too. Like until we're like a few months old, we basically look like fish. So there's not much difference. Like we have a spinal cord, we have sensory nerves, whatever, right? Anyway, so biology figured this out. And I was like, I want to know how biology does it because those are the machines of the future. Like if we can make biological machines, imagine. Yeah, anyway. So I got this scholarship. I came to New York. I did a PhD in biophysics, which is basically like easy biology for physicists. And then I came to Boston and I did a postdoc, which is just like, I thought it was awesome. I got to work in a lab and they were like, what do you want to do? And I was like, well, I have this crazy idea. And then they let me do it. And then that allowed me to start my own lab here at Salk. And so I started here, moved here with my beautiful wife, who is also in the audience. Say hi, Becca. I met her in Boston, so go to Boston, it's a good place. So anyway, here, I'm so happy to be here, and over the next couple of minutes, I'll tell you about what I think is the future of biological engineering. And it's specifically this method called DNA origami. Basically, you'll find out about it soon if you don't know what it is already. This is the question I ask. Well, what if we could use DNA as a building material? You might think, well, isn't DNA already around? And it totally is. But I would argue that it's been overlooked in one way. Like, it carries information, but it doesn't really, like, do stuff. But what if we could make DNA, like, build DNA into things and then have them do stuff? That would be, then maybe we would have biological machines, or, like, the beginning of it, at least. So I'll tell you this story in 3 chapters. So first, I'll try to convince you why DNA is, like, a great material to build things out of. I'll also tell you who first started thinking about this. It was not my idea. I'm glad to be making some contributions to this field, but I'm like, every scientist stands on the shoulder of tons of other lesser-known scientists, oftentimes. And now I'm like the lesser-known one, but that's still cool. So I'll tell you about the history of the field, and then some of what I think are really promising examples of what we can already do with DNA, and why I think this could be like a really cool direction to go in, not just because it sounds interesting, but also because it could actually like change the world. All right, so the use cases I'll tell you about are virus traps, microchips, and molecular engines. These are just 3 crazy ideas that actually work. All right, so DNA. Let's start with DNA. So hopefully you've all heard of DNA. You have cells in your body, and the cells have nuclei, hopefully, and then inside the nucleus you have a bunch of DNA. The funny thing is, like, when you see these pictures, that's not actually what your chromosomes look like. That's what they look like when the cells are dividing, and you like separate the chromosomes from one another. But most of the time, it's kind of a blob. In the nucleus you have the DNA, and the DNA itself is a polymer. A polymer is just like a long string of molecules connected together. In this case, the DNA has 2 strands, and they're spiraled around each other. And then every strand has a sequence of what we call letters, and those letters could be A, T, C, or G, and they pair together in specific combinations. And if you look inside the cells, pretty much all the DNA you have is in like a perfectly complementary duplicate. So you have like some sequence on one of the strands, and then the opposite sequence on the other strand. So for every C here, you have a G here. For every A here, you have a T here. And that holds it together in this kind of spiral conformation, this helix. So that's what it looks like in your cells. But in reality, you could like peel this apart and have like 2 separate strands, right? And those 2 separate strands could have any sequence. It just happens to be a particular sequence in each and every one of you. And for billions of years, like since life came around on Earth, which we don't really know how that happened, but since it began, it seems like there's been DNA in all of the cells that we know of, and the DNA, carries information for how to build life, essentially, like how to make RNA and proteins and then cells and animals and plants and everything. And so that's been kind of the use, right? Like in nature, DNA stores information. We keep hearing this, like, oh, DNA, it's the blueprint of life, blah, 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 right? And that's definitely true. And it's for a reason. The reason why DNA is such a good thing to store information in is because it's programmable. You can change that code, and it has like the same structure, same functions, same shape. Like it doesn't change much. You could just like alter the letters, and you can have the same DNA but with different instructions, right? It's very stable. Like maybe you've read that sometimes they find these mammoths like buried in the ice, and the mammoths, like they're tens of thousands of years old, and the DNA is still relatively intact. And so now we know the exact genomic sequence of mammoths, and some people are actually making them over at Harvard, mostly because they like furry, big animal pets, but also I think they think it's good for climate change or something. I'm not sure. But anyway, it's really stable. If you bury like DNA in the ground, it will eventually be eaten by other animals, but if you don't eat it, it doesn't go away. It's super stable, which is great. It's also easy to copy because you have these, if you have a sequence, and you peel it apart from the other strand, then you could just like find which letters match, and then you can copy the DNA sequence. That makes it's like a method to copy the information that is used by all of your cells, and it's easy to duplicate. I would argue that for the exact same reasons, DNA is also a great engineering tool. So first of all, we understand how DNA works. Like a lot of scientists have been working on DNA for a long time. Now we know a lot about it. Not everything, but quite a lot. Compared to other molecules, like we actually understand DNA pretty well. We can program it to have any sequence. That will come in handy soon, you'll see. It's cheap to make. Thanks to this whole like biotech revolution, like now it's actually really easy and cheap to just like make large quantities of DNA of any sequence. It is, as I said, easy to copy. It's resilient. It doesn't break down unless you eat it, in which case it's actually not toxic. I mean, most of the things you eat is just like tons of DNA, and it just like becomes you, right? And it also works in biological systems already. So if you wanted to make like replacement parts for humans, for instance, maybe we should make them out of DNA. Like that's kind of already biologically compatible, right? Okay, so I'll tell you where these ideas came from originally, and then I'll tell you where we are now. So it all started with these fish. These fish were part of a painting by M.C. Escher. And so M.C. Escher was this guy who just like made weird patterns. And these fish are part of a painting that is quite famous. And it was hanging on the wall of a scientist in New York in the 80s. And this scientist was studying like crystals and chemistry in part of DNA. And one day, he looked at his painting, and he was like, why does DNA have to be this double helix? What if it could be other shapes? What if I could make DNA into a little fish with 4 fins? This is a true story. I met him, Nedrin Seaman. He unfortunately passed away last year, but he's like a legend in the field. They kind of look like these fish. In the beginning, he thought these would be useful as like a framework to build like matrices, like large kind of framework structures. And you can see how it's built. Like that structure is pretty straightforward. And he actually wrote this up in a paper in like 1982, so before even I was born. Basically, it's such a simple idea. He was like, if we could create DNA with these sequences here, like GCACC or CGAGT, right? And then we made these 4 pieces of DNA. They should come together like this, right? They should just like find each other and then pair together and create this cross-like structure. It was just a hypothesis at the time. Like you couldn't actually make DNA really well back then. And he wrote this paper in the Journal of Theoretical Biology, which even back then wasn't really read by many people. And very few people really paid attention to this, but Ned was undeterred. He spent the next 10 years trying to make this happen, and he realized that nobody would be impressed by little cross structures, so he was like, what if we make a cube? And this is basically a bunch of cross structures. I mean, they're Y-shaped junctions, but similar idea. So basically, he came up with this structure, and he was like, what sequences do I need for this thing to come together and make a cube? And then he spent a lot of time making those sequences, and then he spent an even longer time trying to show that he had made little cubes, because these cubes were tiny. And so that was his proof, and it was published, and now people started paying attention. But it was very unconvincing that this was going to be useful for anything, because you see these like little bands in a gel. It basically just shows you that it has a different structure from the other pieces of DNA that he made. And so back then, the limitations were, this was really expensive to make. It was difficult to see what you had made once you made it. And it was like very few of these cubes actually became real cubes. Like we now know that most of them didn't even become cubes. But since then, we've gotten a lot better. So now we can build DNA like Legos. There are companies like Twist Biosciences that you just literally go on a website and you type in which sequence you want, and you can like order DNA with any sequence, and it just costs a few dollars. And then you get a test tube with like that DNA with that sequence. We can also look at the DNA structures if we make them using atomic force microscope or transmission electron microscopes. And this is what some of those early structures of DNA look like. So a tetrahedron or cubes would look like that too. And then the big leap forward came in 2006. There was a guy at Caltech who basically came up with this method, and he came up with the term DNA origami, which I actually think is maybe the biggest invention, because now people were like, oh, that actually, like, it makes sense. Like, I can fold things out of DNA. That's cool. So the way we do it now is we take a scaffold DNA. So this is a long-stranded, single-stranded DNA. And then we combine it with these short pieces of DNA that we just make in like random, or not random. The scaffold DNA has a random sequence. It's something that we take from nature. And then the staples are short oligonucleotides, like short sequences of single-stranded DNA that we create with specific sequences to sort of pinch together the scaffold in different ways. So if you look here, like this staple strand here might be complementary to this part and this part of the scaffold DNA. And so if it binds, it will like bring together those parts of the scaffold DNA. And if we're clever about it, we can make the scaffold come into any shape that we want. So the first thing that Paul Rothman did was so basically now we're overcoming all of those obstacles. And the first thing that Paul Rothman wanted to make was a smiley face, just to show that he could make literally any shape. So he was like, okay, if this was made of DNA, it would basically be a bunch of DNA helices, and then I want to like stitch them together in this direction. That's like a montage for how I made it. So he was like, okay, I need to order a bunch of DNA with different sequences, and they need to be complementary, and then he mixed them together, and this is what it looked like. This is like actually made of DNA, right? Imagine like looking in your microscope and then like scanning a surface, and then this thing looks back at you. Like that must have been so wild. The crazy part of this story is that Paul Rothman did this by himself. Like he didn't even tell his students. He was a professor at the time. His students were like, we don't know what he's working on. He's like always walking around in the hallways like mumbling to himself. And great guy. But like kind of weird in the best of ways. Has these awesome ideas, right? And then he published a paper with himself as the only author, showing that you can make smiley faces out of DNA. I mean, like, yeah, I think that's legendary. Anyway, that made people like me realize that, like, wow, maybe this, has a future. Maybe if we can make smiley faces, like, imagine what else we can make. Like, maybe we can actually make useful things. But before we make useful things, let's make more emojis. Some other people at Harvard realized that you could fold these structures in 3 dimensions. And so instead of making these flat emojis, maybe we can make 3-dimensional structures like blocks, or even curved structures or components to machines. So these are just computer renderings, but all of these pieces you see here have also been made in reality, and they've used electron microscopes to make sure that they look the way they do. So now we have this like new tool where we can build any shape we want out of DNA. And we even have apps to help us design these structures. So we can sort of like design a three-dimensional structure, and then using these softwares that I'm happy to teach you guys if you want to learn how to use it, you can learn like which sequences do I need to order from these companies. And then you spend, well, maybe like a couple $100. Depending on how complicated your structure is, it will be like tens of dollars to hundreds of dollars. But then you order these components, they show up, and then you mix them together, and you heat it up a little bit, and then you cool it down, and then out comes structures. So we thought, why not make a Nerf gun? Well, actually, Mark Rober thought that. He's a YouTuber, and he made the world's largest Nerf gun, and he was so excited about making more Nerf guns. And he approached me, and he was like, I heard you guys, like, you work with DNA. Could we make, is this something you could do? Like, could you actually make a Nerf gun out of DNA? And I said, well, we can make the shape. We can't make it shoot quite yet, and I also don't know why we would do that, but I'm happy to make the shape, at least an outline. And so I talked to one of my students, and I was like, Lauren, can you make a Nerf gun? And she was like, of course I can. And so she designed this structure, and those are just DNA helices. And then the way that we like order these components, right? And then hopefully we hope that they would sort of find each other in the test tube, in the solution, and then come together into this predetermined structure. And so I'm showing you like how we think that a part of this is folding. But if you zoom out, this is actually just like the same thing over and over again in a particular structure that then forms this shape. So it's no different from the emojis that you saw before. It's just that now, we made a Nerf gun. So that's cool. That's what it actually looks like in reality. And Mark Rober was impressed, so we were happy. You should check out the video. That was super cool. Just to give you a sense for how big this is, we had an illustrator draw, like, for scale, what would this look like? So here's an actual, I should have brought it. It's in my lab. I can show you later. But a Nerf gun is like this size. But if you zoom in on a penny, this is how big a penny is. If you keep zooming in, that's the size of a hair. You keep zooming in, like that's what one of your cells look like. They have different sizes, but that's like a skin cell. You keep zooming in, that's a chromosome. And then the powerhouse of the cell, right there. You keep zooming in, that's how small the DNA origami blaster, knock on, look like. And yeah, so it's pretty cool. We made like trillions of these for a couple of dollars, and I thought that was pretty cool. So hopefully more people realize that this can actually be used to build like literally anything. And yeah, in the future, I mean, we had, this ended up becoming kind of a thing, and I had all these discussions with Mark, and he was like, well, but can you actually make it fire? And I was like, why? And he was like, well, just think about it, you're a scientist. And I was like, well, yeah, I guess we do fire things on the molecular level. Like we all got vaccines, right? Like Jonas Salk designed the vaccine for polio, and you have to kind of like shoot it into yourself, right? And then once the vaccine gets into your body, it also has to go into your cells. Like the COVID vaccine, for instance, it's an RNA vaccine. Those RNA molecules have to go into cells. So maybe we can make it more efficient if we actually like shoot the vaccine into the specific cells that we wanted to reach. So then we had this whole discussion, so now we might actually make a firing DNA origami Nerf gun. Stay tuned. All right, I'll show you some actual examples of what this could be useful for in the near future. So any ideas what's going on here? Huh? What are those things? They're cells. What are those little white dots? Any ideas? Huh? No, they're not organelles. They're way worse than organelles. They're viruses. Yeah, that's COVID. That's COVID infecting human cells. And the human cells, if you can't tell it, they're not doing too hot right now. They are, yeah, they're kind of like, some of them are blowing up, some of them are fusing. They're just generally unhappy. If you compare the beginning to the end of the movie, there are way more white spots now. And that's because COVID is essentially reprogramming the human cells to make more COVID. Oh, look at them. Not happy. Yeah. Yeah, you don't want that. Get vaccinated, everyone. This is obviously bad. Sometimes our immune system can clear the infection. Sometimes we have vaccines that prevent us from getting too sick. But sometimes people get really sick and they can't clear the infection of vaccines, of viruses, even if you have vaccines. And then in those cases, we need help, right? So there was a lab in Germany that realized that what if we could make little traps for the viruses? We could just build them out of DNA. And then these traps could actually encapsulate the virus particles and make them harmless. So they built this spherical shell out of DNA origami and then made the shape of the shell exactly such a size that it would fit COVID viruses, like the SARS-CoV virus particles. And because that's pretty much the only thing of that size in your blood, these shells should only pick up those viruses. And then they could like coat the shells on the inside with virus-sticking peptides. So here's an actual experiment. These are cells where the cells themselves are not green, but they made a version of the virus where when the virus infects the cells, it turns the cells green. So then the cells that are green are infected with viruses. This is if you don't deliver these shells, this is what happens to cells infected by virus. And then if you add drugs against the virus, like antibodies, you see fewer cells get infected, and they're less infected. But if you add these virus shells, now very few cells are infected. And here's a quantification. So this is just from like cells in a dish. So this is obviously not the same thing that happens in people. But these little shells are now being explored as a potential therapeutic to cure virus-related diseases. So I think that's quite promising. Okay, example number 2, circuit boards. So circuit boards are what computers are made of, essentially. It's just like a bunch of wires that are laid down on a piece of plastic. And then as time went by, we needed to make more wires in a smaller space, and then the plastic chips turned into silicon chips, and then the wires turned into just like little tiny imprints on the silicon chips, and now things are really small. And so in order to make the chips that we have in our phones and in our computers, we need to make really small wires that are in specific directions. And that's hard. And we're actually pretty much at the limit of what we can do. So this lab in Boston had this idea, what if we make little shapes for where the wires are going to go, and we make those shapes out of DNA. So they made these little landscapes, essentially, out of DNA origami, and then had attachment handles on these little landscapes, like here. And these little attachment handles, they bind to the cables. The cables here are just carbon nanotubes, but that's basically little cables. And then the carbon nanotubes would lay down exactly in the direction and geometry that we designed, not we, that they designed in these landscapes. And then they can create microchips or nanochips that are way more precise than what you could do with other methods. And once you've built this, you can even like remove the DNA and the wires will still be there. So maybe this is how we'll make computers in the future. Yeah, here we go. And then the last example I show you is something we made in my lab. We made arguably the smallest human-made biologically powered engine. So to do that, we took inspiration from nature. I thought, in biological machines, we need moving parts. So everyone else kept making these structures that are just standing still, and that's nice. You can build components, virus shells, scaffolds for microchips. But if we want to make machines, we really need things that move too. And it turns out that our body already figured out a lot of this. So if you look inside of our organs, there are cells. If you look inside of cells, there are nuclei. If you look inside the nucleus, there's chromatin. Chromatin is just a fancy word for DNA and proteins interacting with each other. And so there are a lot of proteins in your cells that interact with DNA. And some of these proteins, like this one over here, they actually rotate the DNA. So I thought, what if we design a DNA origami propeller? We also need a way to see it, so we put some fluorescent markers on it. And then we can connect this to a biological motor. Then maybe we can use ATP, like some biological fuel, to create a moving component. So this is what the rotors look like, the propellers. They look okay. If you zoom in, you can see that they're correctly folded. They have the shape that we designed. And I think they're super cute. And then we connected them to these motors. So you can also just like order these motors online, turns out, which is very convenient. So we got these motors. This one is called RNA polymerase. It actually came from bacteria, but we have similar ones in humans. And then we connected it to a rotor, and there's just a regular piece of DNA like connecting the rotor to the motor here. And then we fed it with biological fuels, so nucleotides like ATP. And this is what we hoped would happen. But we looked at it in a microscope. The pictures you saw from microscopes previously were static pictures. They were electron microscopes or atomic force microscope. You need to kind of freeze the sample in order to see it. So that wouldn't be good if we wanted to watch moving things. So here we had to use an optical microscope to see things that are happening like in a solution live. And these propellers are really small. So it basically, you saw these are the pixels on the camera in a microscope with a super high power objective. And even then, you can barely make out what's happening, but we actually traced the movement of the light, and it moves. That's the trajectory you see there. And yeah, so this was the first example that you could actually power these propellers using biological fuel, and then you can see the steps. So here's like rotation on the y-axis, time on the x-axis. You can actually see the steps of this motor using one molecule of energy at a time, like one ATP at a time. And I think this is the first step to making actual functioning machines out of DNA origami. So where do I see this going? Well, we have a lot of projects underway in the lab. We're building sensors. We're building devices of different sorts. Hopefully we can make like energy generators, turbines, all sorts of things. But this is kind of limited by our imagination and how hard we're willing to work on this. So I want to make an analogy and then kind of pose a challenge to you guys. So if you look at a timeline and you think about another technology that has been around for a while, the transistors. So does anyone know what a transistor is? Kind of. It's what microchips are built of. So the wires on the microchips, they connect transistors. The transistors are like the fundamental unit of computers. They were invented in the 1950s. And it was a bunch of nerds in Bell Labs, which was a science institution. They were like, wow, if we connect these three components, it has really weird properties. Like you can gate whether or not the current will go from one side to the other. And it was just kind of a curiosity at the time. It was a science experiment. But they had a vision. They were like, if we can connect these things together, we can actually compute things. Like we can actually perform logical operations. A little bit similar to how our brain works. Not quite. That took a long time before we figured that out. But back then, they had this vision that like, if we just make these things more complex and develop how we build them into structures, maybe we can do something useful with it. And then for like the next 50 years, very little happened to the regular people in the world, right? Like up until the 2000s, computers didn't really play a major role in people's lives. Maybe in the 90s, fine. But before then, absolutely not. And it wasn't until we could put billions of them together in really complex chips, and there were software engineers that made software programs out of it and apps. Now all of a sudden we have smartphones, computers, self-driving cars, like all the technology that you guys take for granted was built on 50 years of nerds working on transistors. And if you really paid attention back then, you also would have realized that this thing has potential. We just need to put in the effort to make it into useful technology. I would argue that's where we are with DNA. DNA origami, the first vision came around 40 years ago in the 1980s. And since then, sure, we made smiley faces, we made little cubes, we made, cute structures. And maybe now people are starting to see that we can make useful things out of DNA. And if you think about this timeline, we're about maybe 10 to 20 years behind transistors. It's up to you to figure out what we do with this technology and where we go with it in the future. So to help us figure out what that would look like, I asked ChatGPT to envision a future built out of DNA. So, I think it's cool. This is what transistors can do. Like, if you actually want to build a future, you guys have to do it. And I hope you will use DNA. Thank you for your attention. Thank you to my wonderful lab who makes all of this happen. I'm so happy that I get to work with them. And I hope you guys will make way cooler things out of DNA than I ever dreamt of. Thank you very much.
students, if you have, I don't know if this mic is on, but students, if you have questions, if you could go to the microphones that are placed up towards the top and down at the bottom. We are recording this, so we want to be able to capture your questions. So if you feel comfortable,
But no pressure at all.
full pressure.
Just like get up on stage and
Just talk in front of hundreds of people. Or you can raise your hand and we can just repeat your question into the microphone. So any questions? Yeah.
So you were saying how, sorry, I'm losing my voice. You were saying how you want to make Nerf blasters and like shoot things, and you made like basically perpetual motion machines, so they're constantly going. But in order for a Nerf blaster, you would need the blaster and like the bullet, so that would be separate, and then you have to trigger it at a specific time, like only once. So how would that work exactly? Or do we not know yet?
I want you to figure that out. I feel like I can come up with an idea. This is the thing, right? Like we have the tools. Like I've worked so hard, and so many other people have worked so hard on getting us to a point where we have the tools to solve problems. But like solving the problems themselves, that's kind of the fun part. Like I can tell you how I would do it, but then like my students can come up with their own ideas. So you named 3 things, right? You need the blaster, you need the bullet, and then you need the firing mechanism, right? And it has to be like a one-time thing, right? So Nerf blaster, done, okay? Bullet, it could be whatever. Like let's just take a little, we could make it out of DNA origami. I mean, seems like a reasonable thing to do. You can, if you want to do like gene therapy, for instance, you can literally take a gene, fold it up into a little packet, and then that's your bullet. Cool, you can cure genetic diseases. And then firing mechanism. That's the crux. That's going to be the hard part. So how does it, do you know how an actual Nerf blaster works?
Yeah, I watched the video and I saw him make it out of a single piece and like bending it and
Yes.
storing the energy.
Exactly. So how would you do that out of DNA? What's an example of like storing energy?
Spring?
Yes, DNA spring, right? So I've worked with some scientists in Germany, and they came up with a way to actually, the first guy to do this was actually here in California. It's one of our collaborators. He literally took a long piece of DNA. It was like way longer than any of these things and what we would put in a Nerf blaster. But he took a long piece of DNA, attached it to a magnetic particle. And magnets, if you know, like if you put a magnet next to another magnet, they would kind of align. And then if you rotate one of the magnets, the other magnet will also rotate. So what he did was he attached the DNA to let's see if I have a cable I can show you, to like a surface and then to a magnetic particle. And then he held the magnet above it, and then it like stood up like this, and you could see it in a microscope. And then he rotated this magnet, and this DNA started rotating, and he realized, oh wow, I have to rotate this for a while to notice anything. So he connected it to he went out and bought a Lego like an engine motor thing, and then he just like spun it really fast, and then eventually this magnetic particle stopped spinning, and that meant that the DNA was wound so tightly that it was like resisting more spinning. And then he removed the magnet, and this thing just like spun back. That proves that you can store potential energy, spring-like energy, in DNA. And so if we can twist DNA to store energy, maybe we can convert that spinning into push somehow. So that's one way. Another way is maybe you can store energy by compressing things, right? Then maybe you could build a structure of DNA origami that is kind of like a wire frame, and then maybe you put a magnetic particle or something to like compress it, right? And then you would have to have another DNA piece that like locks it in place. But then you can remove the magnet, and now you just have a DNA origami structure that has that stored energy, and you need to have a trigger. That was the fourth part that you didn't talk about, but I think it was kind of included in the firing mechanism, right? So the trigger itself, that is actually relatively easy. You can make DNA pieces that break apart if you shine a certain wavelength of light on them. And then you can do it with light. You can have it like respond to a chemical signal. So for instance, I have these engines, right, that respond to chemical signals, but there are lots of ways that our body responds to chemical signals. We could just put one of those things in the DNA. Or maybe they can respond to, let's say that like viruses have like RNA in them or DNA. So what if like when that RNA binds to the Nerf blaster, it will like trigger the release, right? So like maybe that thing will like bind to something, and when it binds to something, it releases it. There are many ways to think of it. Anyway, great question. I would love to do it. We just need more smart brains to like come up with the specifics. It's totally doable. Like it's totally doable. Yeah. Anyway, Mark might pay for some of it.
To add on to like the motor that you made, is it possible to like combat climate change and stuff and like someday make like a motor that like powers cars and like trains and stuff? Like, is that possible to like do that?
So you mean from the, I mean,
Like DNA powered cars.
well, how did you, you walked from your seat up to the microphone. That was literally biological motors doing it. And it wasn't one motor. It was like trillions of tiny motors within your muscles doing this, right? So yeah, one of these DNA motors could probably not power a car. But if we could figure out a way to combine them and have them work in series or in parallel, yeah, you would need trillions of them to do any measurable thing. But that already happens in all of us all the time. So specifically for a car, again, it's like, how do we solve this problem? I don't know how to make it move a car. I would actually say just like putting things together, it's easier to combine things that are moving in a linear direction than to combine things that are moving in a rotational direction. So that's probably why your muscles work, because you have these motors that like pull on filaments. So your muscles have these fibers, and then these fibers that basically like pull on each other like this. So they slide relative to one another, and that's what contracts your muscles. So if that I can see how you can do. Basically what you can do is you can, instead of having one of these rotational motors, you would have a motor that pulls the DNA in this direction. We have those too. And then you would just have a lot of those in like build them. You could build a scaffold out of DNA origami that has like organizes a lot of them in parallel, and then it would just pull things like this, right? And that's, if you think of like a steam engine, that's how a steam engine works. It actually, you don't know what a steam engine is. That was probably before your time. Back in my day, in Sweden, when we took the steam engine to work, to school, a steam engine actually like pushes a piston and then pulls it, and then pushes it and pulls it. And then there's like this thing that converts that into a rotational motion, like this. I don't know if you've seen it. So it's just like a bar that connects the piston to a wheel, and so it like pushes the wheel back and forth. It's kind of similar to how a bike works. Your legs push up and down, but then the bike turns that into rotational motion. So that's how I would solve it. You guys are asking me to solve all these problems. Like you're the smart kids. Like, sorry, not kids. You're adults. Are there adults?
Not quite yet.
Not quite yet. Okay, you're smart kids. Yeah, thank you. Great question, though. I do think we should parallelize this and make them into useful, larger structures. Yeah.
So I know that you can ask like other companies to produce like DNA origami.
Yeah.
Can you patent like specific structures that you make, or is it like posted under like Creative Commons? And if so, how do you properly like cite the DNA origami or structures that you use from others?
Okay. Wait. Can I just ask you to clarify? Are you asking about the patent because you want to make money off of this, or are you just wondering like how we like spread the knowledge? Both are valid questions.
Both.
Both? Okay.
We will have a patent attorney with us later on during our career panel.
Oh, cool. Yeah. So patents get a bad rap. Like I think patents. Yeah. A lot of people get super rich from patents, and it's easy to focus on like, oh, these people make money from patents. Like we shouldn't have patents. Jonas Salk didn't patent the vaccine for polio, and I think that was an awesome decision that made it free and available to the world. All of the things that we do in my lab will be accessible to the world. Like we will just post them on like servers and in publications and whatever. We might patent some things that where if we think that it can do a greater good if we get investors, then it might make sense to patent, because that way those investors will know that their money will be protected. Like basically that they're not going to be, like somebody else isn't going to make the same exact thing. But we would only do that if we really think that this pathway is the best pathway to actually spread the technology and do the greatest good for it. So that in some cases that's true. In other cases, it makes more sense to just make it openly available and have like regular people make use of it. But actually, for these things that are really far from applications, like they still need a lot of work before you're going to see these things in people's houses, right? So you do need to put a lot of money into that development, and if you're going to do that with like taxpayer dollars, like we use a lot of grant funding, right? Like from government agencies. That is like not as much as the money that some of these big companies have. And so if like Google, decides to develop this as a product, they could actually make it happen way quicker. And that's why in some cases I think it's great to have a patent, because if you have a patent, you accomplish both. Like the company can develop it, but the patent itself would actually be published. So after a year after you file the patent, it will become public knowledge. Anyone can read it. Like you can't start a company and make the same thing, because then the other company will sue you, right? But if you make it in your house and you just use it for personal use, like that's fine. Usually if universities use it, we write the patent, right? So we can write it such that if you're doing this for a nonprofit purpose, you can totally use our technology. And in that case, patents are super useful because they're basically a publication, right? So yeah, you can do both. We're gonna make sure all the research we do at Salk, we want to do it in the spirit of Jonas Salk. We just want it to be as beneficial to people as possible. Yeah.
You were talking about the happy faces that were made, and I wanted to ask a question about possibly using this as a biomaterial, where if you have the happy faces, could you break the DNA, or is it flexible, and then could you use it as like an alternative to carbon fiber?
Oh, that's literally what, who's doing that? Lauren. That's what Lauren is doing. Lauren is starting to figure out how flexible these things are. Yeah, turns out we don't actually know. People were so obsessed with looking at what they look like that they didn't bother to check what their material properties were. And so we are as well, not as we speak. They're probably have a weekend now, but that is what we're measuring. We're trying to, and it turns out it's not that easy to measure the material properties, because you have to kind of like grab it and twist it, and we're figuring that out. It's slow work. But what we think is that it is about, I mean, it's like, it's way more resilient. And depending on how tightly you weave it, like how tightly you stitch it, you can make different properties. You can make it like more rigid or more flexible. And you can, it's easy to make it more flexible because you just like make little holes in it, kind of, and then you can make it more twisty and bendy. And we think that could be good for like creating like patches that conform to something. Like if you want to create like an artificial, like scar tissue or coating or something. But then if you want it to be more rigid, like if you want to make machine parts for devices or engines or motors, you would want them to be more rigid typically. And so then we can make them more compact. And then we've seen that they are more rigid. There's the lab at MIT that is making a simulation software where you can try to calculate the mechanical stiffness. But for now, those are just numbers. Like we have to also make measurements to see if those numbers are correct. But those are built on like physics knowledge of DNA, which is so-so. Yeah, but it's a great idea. I mean, I think this could be used for, like if you're creative, anything you use, like plastic. Plastic is only, I don't know, 70 years old or something. Before then, there was no plastic, and plastic is a polymer. And now we can make plastic of all different sorts of hardnesses and flexibilities. And plastic is used in so many things, So I think DNA could take the place of plastic in many cases, and it's way, like you can eat it, right? It's nice. So, yeah, thank you.
So we're going to go to the gentleman in the yellow sweatshirt. He's been waiting a little bit longer.
Oh, okay. So you showed us that these German engineers have created a trap for the COVID-19 virus. And my question would be that, can these traps also encase the many other different types of human known viruses? Like, for example, can you put the AIDS virus in these traps?
Yeah, that's a great question. That is the vision. So when they developed this technology, COVID was on everyone's mind. So it seemed like the most urgent thing. I don't see any reason why not. I mean, the technology is two components. It's making the shell to the shape of the virus, and the other one is making the inside sticky to the virus. Okay. So as long as we can solve both of those, it should be possible, yeah. It should be possible. There are obviously hurdles. So for instance, your body is scared of things that aren't your body, right? So if you inject people with DNA origami, eventually your body will be like, foreign object, let's fight it, you know? Like, I'm allergic to nuts, and nuts are totally not going to hurt me, but my body still thinks they're dangerous, right? So my body will for sure think that DNA origami is dangerous. But this is also just an engineering problem. So there are people working on how to coat these things so that they're kind of invisible to the immune system, basically to make it like not be bothered. But if we can do that, I think that is the biggest hurdle, like just making your body not reject it. Then it should be fine. Because once the viruses are encapsulated in these origami particles, it's like totally harmless. And there are no, like, aside from the immune reaction to the origami, there doesn't seem to be any negative effects to these things.
Then would the age of viruses cease to exist? Because now, like, for instance, we have now encapsulated every known human virus.
That's a great question. Well, I'll ask you a counter question. We have antibiotics, right? Mm-hmm. Have bacteria ceased to exist?
Not really.
No, they come up with solutions, right? Every time we come up with a new antibiotic, there's 1 out of 1 trillion bacteria that has some mutation that allows it to escape, right? And that becomes the next generation of bacteria. I think the same thing will happen with viruses. And in a way, I think we should think of this as an ongoing competition, not as like a fight to win, right? If we do, that would be nice, probably. Maybe viruses have some beneficial effects too, but probably not AIDS. But for bacteria, right? Like we just have to keep up with it. We don't want to die from I don't know, like strep throat or something. That would be bad. People used to die from bacterial infections all the time, like tuberculosis. And nowadays, we have great antibiotics to treat them, but there are still some diseases that can overcome the antibiotic resistance or that can resist the antibiotics. And we just have to make sure that we keep pace with the bacteria. And so I think this is it's like a new tool in order for us to help fight virus diseases. And maybe people don't have to die from AIDS. But I think it would be hard to eradicate all viruses based on this. Yeah. And also AIDS, AIDS is really tricky too. AIDS, I think the reason why we haven't really gotten rid of AIDS is because it's one of those viruses when it duplicates or when it reprograms cells to make more viruses, it does that very poorly. So every time a virus infects a cell, the viruses that come out of that cell are quite different. So if we make a virus shell, right, that is a certain size, maybe the viruses that come out of one cell have a different size, and then HIV has overcome our defenses. But then we just have to be more clever and outsmart the next generation of viruses, right? I think that's unfortunately the way we live. I will say though, now that I'm on the podium and I can tell you my opinion, I think viruses are underestimated. Like we see them as these like bad people, right? Like, but like that's how we used to think about bacteria too. And then some people were like, well, maybe bacteria are not all bad. And we realized that for us to be healthy, we need to have a healthy microbiome with a bunch of bacteria that work with us in our gut. And now we know that they have bacteria everywhere in our body, and a lot of them are actually beneficial to us. I think the same might be true for viruses, we just haven't recognized it yet. Obviously not all viruses, but some might actually be good to have. So we want to be careful before we eradicate everything of a certain part of the ecosystem.
I see.
Thank you.
Thank you very much.
I had a related question having to do with some of the cancer research too. Is it possible that DNA structures could possibly limit the mass cell division that takes place because of cancer mutations and such? Is that something that's not just virus targeted, but could also be used to reduce the over amounts of cell division that happens when?
Using DNA origami to prevent cancers from growing, is that what you're saying? Mm-hmm. How would you do that?
I don't know. That's what I'm asking. Is that something that's possible, or is it entirely related to just viruses?
Okay, so sure. I'll give you one example of how I would do that. This is like literally, I guess this is why they pay me, to come up with these ideas. They don't pay me much, but it's okay. I'm doing fine. I'll be fine. Fine, thanks. But it's a pretty cool job. We can make things that kill cells, right? Like, for instance, a lot of cancer therapies are built on recognizing and killing cancer cells. So it used to be, I mean, it still is, that chemotherapy is like how we kill cancer cells. Unfortunately, that kills every cell that divides. So that's not great, right? Like immune cells, the cells that create our hair, all of those cells divide all the time. And so if you take chemotherapy, you'll kill those cells too. Bad, right? The problem is never how to kill cancer cells. The problem is how to kill cancer cells and not kill your own like healthy cells, right? Every time you read a news story that says like, oh, this chemical in tomatoes kills cancer cells, it's like, yeah, well, so does bleach. But it's like not gonna. That doesn't help us. Like what helps us is things that kills cancer cells, but not our cells. So if we can use DNA origami to make more targeted therapies, that would be the solution. The way a lot of people are doing it now is that they're reprogramming immune cells to recognize cancer cells as a foreign or dangerous substance, and then the immune cells will kill cancer cells. This is a super promising direction for science. Some labs here at Salk are actually working on this. It's called immunotherapy or cell-based therapy or T-cell therapy. I think we can make versions of this kind of cell-targeted therapy, but using DNA origami Nerf blasters, for lack of a better term. But basically, if you have a substance that does kill cells, like a chemotherapy agent, and you encapsulate it in DNA origami, and then this DNA origami capsule only opens if it binds to cancer cells, there's your solution. So then we have to come up with a mechanism for how to connect the recognition of a cancer cell with the opening of a DNA capsule. That's a very well-defined problem. And when it comes to targeting the cancer cell, that has sort of been solved by science already. So in these immune cell therapies, we reprogram our immune cells to recognize a specific molecule that is only found on cancer cells. And this is actually a huge part of science right now, is trying to figure out what makes cancer cells unique. Like what are the molecules present on the cancer cell surface that makes it recognizable and different from your body cells. Whenever we find one of those molecules, and there are tons of them, those are called cancer biomarkers or cell surface markers for cancer cells. As soon as we can identify one, that's the thing that you want to optimize this like opening mechanism for. And there I have some ideas. Like essentially, you basically want something that like once it binds, it starts like breaking down the shell, right? Or causing the shell to come apart. So it could be that you have two molecules that like bind to one another and that holds the shell closed, kind of like a clasp. And then if it recognizes the cancer cell, it like switches and recognizes that thing instead because it binds even better there. And that could open the clasp, right? That's one simple mechanism you could think of doing that. But someone has to do it. So if you're looking for a job.
That'd be great, actually.
Yeah.
Thank you.
Thank you.
Okay,
So
we have time for 2 more questions. So these last 2 are it.
Okay, cool.
could we ever get biological systems to create DNA origami? And would that only happen during like mitosis or meiosis?
Oh, great question. Great question. Yeah. Well, so your cells make DNA, right? I don't know if you would want your cells to make DNA origami. Maybe. I would say the way that your cells make DNA is based on the DNA that is already present, right? It just copies it. So it would be relatively tricky to all of a sudden make it in a different sequence. But if you can inject or deliver a foreign like template into the cells, like a gene therapy, for instance, then that could be the template for how the cell then builds copies. Like if the cell copies that thing, it would make DNA origami. I would say that a better direction to go is, are you familiar with RNA? So DNA gets, there are enzymes in your cells that turn DNA into RNA. And then if you basically, if you put DNA into a cell, you can have the cells then make RNA, like many copies of RNA from that DNA in a single cell. When it duplicates, it just makes one copy of all the DNA. But it makes like many copies of RNA from one piece of DNA. So maybe RNA origami is the way to go. Like basically, you would deliver a piece of DNA into a cell using gene therapy or Nerf blasters, I don't know, whatever. And then there are actually many ways to get the DNA into cells. And then you can have the cell like basically make RNA out of that DNA. And then when it makes the RNA, that RNA could fold into certain shapes. In a way, that's kind of what happens. So ribosomes in your cells, those are basically RNA origami. It's RNA folded into a big machine that like does stuff. So it kind of already happens. I actually think that one way to do this more easily is to just reprogram how we make ribosomes, and then you can make ribosomes into other structures that do things. Might not be good to do in healthy cells, but it would be, you could do it like outside of humans, potentially.
Thank you.
Thank you.
Okay, last question.
So you had mentioned the possibility of biomachines.
Yeah.
Would something like a biomachine have to follow the principles of engineering that we already know today, similar to how robotics has its own like rules and structures? Or would we have to rethink the way we engineer things in a new method similar to how nature builds bodies or insects in their own methods?
You know what? This is like such a great question to finish on. The answer is not known. It's up to scientists and researchers to figure out when our intuition as engineers is still true for molecular structures and DNA origami, and when we need new intuition. And in some cases, we already know that our intuition is wrong, and then we need to rethink it. Like for instance, we can't build things like from the top down. We have to allow things to build themselves. Otherwise, we can't do it with the right precision. That inspiration we took from nature, because nature does that, exactly how you said. But other things, like there was a lady earlier who asked about like how to store potential energy in DNA. Surprisingly, to every one of us, that actually seems to hold true, even for DNA. So that's an intuition that we have, like you twist the cord and it springs back, right? That turns out to be true. So we just have to test it and see in which cases it is actually true, and in which cases we need new intuition. And when we need new intuition, nature is a great place to look, because there are so many solutions that are already out there that if we just understand how nature works, we can actually draw on that inspiration to create better technology. A perfect example of this is we studied the brain of humans for hundreds of years, and then someone, actually one of the professors here at Salk, had the idea, what if we make a computer program that imitates how the brain works? And that was the beginning of AI. Artificial neural networks are basically imitating the brain networks, and then that creates a kind of intelligence that we were not able to do with computers without this new bio-inspired intuition. So I think that's true for DNA as well.
Thank you.
All right, thanks.
Okay, another round of applause for him, because that was way too good.